Good afternoon, everyone. Thank you for your time and interest to attend this webinar. I'm Preston, the imaging specialist for PerkinElmer's life science imaging portfolio in Southeast Asia. Today, I'd like to share with you how we can apply PerkinElmer's automated high-content imaging and quantitative workflow to study the biology of different infectious diseases through high-throughput functional and phenotypic assay. I will start with an overview introduction on high-content imaging and analysis, and present to you a number of publications demonstrating how technology can be applied to study viral, bacterial, and parasitic diseases. For those of you who are not familiar with high-content imaging or high-content screening, basically it can be defined as we are using an image-based screening method to measure the biological activities in cells. Shown here is actually a quote from a review on high-content screening article. High-content screening combines the use of automated fluorescent microscopy with quantitative image analysis, and this allows acquisition of unbiased multi-parametric data at a single cell level. We are not limited to any particular applications when it comes to high-content screening, as long as we have the ability to stain and detect the biomarker and protein of interest in our sample of studies. For example, a number of biological questions can be addressed with HCS. We can quantify changes in response of cells to genetic or chemical perturbation, and then identify genes that are required for a specific process. Or we can look at cellular process like differentiation or proliferation, and then using different models of diseases, for example, be it cancer models or infectious disease. And then we can also add different drugs, small molecules, and compounds to do genetic and chemical profiling to see how the cell responds to this treatment. The workflow from high-content screening can be illustrated by this schematic, and it involves different components such as wetware, hardware, and software. For the experimental part, we will see the cells on the microplate and then treat it with the different reagents such as antibodies, small molecules, fluorescent dyes, or with like infectious agent, let's say if you're doing infectious disease study. And then we'll allow the sample to grow over time to see how the cell responds to the added components. And then we can either perform live cell assay or do staining of the different markers. And then we acquire the sample plates with the imaging system. And lastly, we'll perform quantitative image analysis to look at how the cells respond and show expression of the different markers that were investigated during the course of the treatment or in different disease model. As a summary, high-content imaging analysis allows us to analyze small samples as compared to conventional microscopy. It not only relies on the interpretation of individual selected images, but also allows statistical analysis of a series of images per treatment condition. And depending on the number of images per well and the number of replicates, it can greatly reduce bias as well as the human error that could possibly arise from analyzing individual microscope images by eye. It also allows to achieve higher physiological relevance as compared to a biochemical assay, for example, that look at the binding interaction of a compound to its target protein. And it is often lacking physiological relevance because the cellular environment is missing. Therefore, high-content imaging is the ideal technology to analyze complex models like 3D microtissues, spheroids, and organoids. And this method and technology will allow us to gain deep insight into the biology. And we can generate hundreds of readouts per cell. So instead of just analyzing parameters like the fluorescent intensity and the flow cytometry, high-content imaging allows us to analyze the phenotype of cells and allows us to generate a phenotypic fingerprint consisting of a detailed morphological description of every cell in the assay. Now I would like to introduce you to Operetta, the high-content imaging system in the Department of Microbiology. It is capable of doing wide field fluorescent imaging, as well as bright field and digital phase contrast imaging. It also has a temperature and carbon dioxide regulation unit that enables live cell imaging experiments. And it is fitted with objective of different magnification shown here. And it also comes with a robotic arm that automates your plate handling of samples for imaging on the system. Moving on, let's focus on the most important part of the workflow, which is high content analysis. And it is done using one software called Harmony, whereby you can perform image acquisition, data visualization, segmentation, and more importantly, quantification in 2D or 3D. Shown here is typical image analysis workflow. First, you segment the image to identify objects, such as find nuclei, and then next define the cytoplasm to obtain your region of interest. After which, you can start to quantify different parameters such as fluorescence intensity, morphology within the region of interest. And then you classify them into different populations. And lastly, you start to calculate population measurement and statistics. For example, an EC50 curve for cells in a certain cell population. This slide will explain how we can extract numbers from images using the Harmony software, starting with image segmentation of cells treated with a drug chloroquine. First, we'll find nuclei and then the corresponding cytoplasm in the image. This assay is characterized by the formation of spots induced by the chloroquine treatment. So the next step is to detect the number of spots. And for every detected cells and every region inside the cell, we can now start to quantify parameters like the spot intensity, the number of spots, the area, and many more. For example, the cell marked in orange contains 29 spots, and the spot area is 39 micron squares. And such is the list of numerical properties that is generated automatically for every cell in the image. And this also can be applied to every cell in the assay or screen. Knowledge can be gained by comparing a treated samples with an untreated control. It is clearly visible that the untreated cells contain less spots. And by running an automated image analysis, this can then be quantified. Now we can see that the treated samples contain 5 times more spots than the control. So the treatment obviously leads to spot formation. If there is a serial dilution of the compound being used for treatment, it is not easily possible to generate a dose response curve and determine the EC50 value. As being experienced with the current COVID-19 pandemic, and formerly seen by viral outbreak from Ebola, MERS, SARS, and Zika, infectious disease could also be caused by bacteria as well as parasitic pathogens, and they pose major burden to global health, and without effective vaccines or antimicrobial treatment, it could be devastating. To fast-track drug discovery, development, and clinical trials in response to these diseases, pharmaceutical companies and medical researchers are focusing on 3 key intervention strategies, which are repurposing of existing drugs, development of new therapeutics, or vaccines. So in the next few slides, I will illustrate to you how we can use high-content screening as a tool in drug discovery and research against infectious diseases. Drug repurposing is a strategy to identify novel uses for approved or investigational drug outside the scope of their originally designated purposes. This approach offers several advantages over de novo drug development. First and foremost, the risk of toxicity is much lower, as repurposed approved drugs have been proven safe for human use in the original indication. And drug repurposing has the advantages of identifying potential treatment on a shortened timescales, such as in response to the current COVID-19 pandemic. In this paper shown here, the author took advantages of a high-content screen of over 3,700 compounds at different stages of clinical development to identify an FDA-approved compound that reduced mucin-1 protein abundance. Elevated mucin-1 level predicts the development of acute lung injury and the acute respiratory distress symptoms, and correlate with poor clinical outcome. From the screen, the author identified fosmanogepene and proposed as a candidate for the treatment of acute lung injury. Through further characterization using PerkinElmer high-content imaging analysis systems, the author were able to verify that the FDA-approved compound R406 can control the expression of mucin-1 protein as indicated by the decrease of mucin-1 staining in the increasing dose of the compound treatment. So from these studies, it provides a rationale for pursuing clinical trials to test whether repurposing of this FDA-approved drug may have salutary effect for patients suffering from acute COVID-19 lung injury. In recent history, another virus gaining attention was the Zika virus. Zika is a flavivirus that is primarily transmitted via Aedes mosquito and sexual contact. Maternal Zika virus infection during pregnancy also has the potential to result in significant birth defect. There are currently no targeted therapeutics for its treatment, nor there is a vaccine, and HCS was used as a study that aimed to identify novel inhibitor from its library of 774 existing FDA-approved drugs that could potentially be used for the treatment of Zika infection. So in this paper, the authors screened a library of more than 700 compounds for anti-Zika viral activity, and more than 20 compounds were identified to exhibit antiviral activity, and they selected a few compounds for further characterization in multiple cell types. Using an immunofluorescence assay measuring the Zika viral envelope protein expression after infection, the several drugs shown here were able to reduce Zika viral infection in primary cell line derived from the placenta tissue. So shown here are example images whereby treatment with one of the drug, MPA, significantly reduced viral infection as indicated by the decreased viral staining in red. For comparison, the positive control NITD-008 and negative control DMSO were compared respectively. This study identified drugs that could be tested in clinical studies of Zika viral infection and provide resources of small molecule to study Zika virus pathogenesis. In the last example, we look at how high-content screening and imaging can be applied for the discovery of small molecules that interrupt malaria transmission. In this paper, the authors developed a serum-free one-step assay for malaria transmission blocking activity, and a total of close to 14,000 known and new compounds were analyzed. Over 90% of these drugs did not show activities against the late-stage gametocytes, which are metabolically quiescent. However, several compounds with low nanomolar transmission blocking activities were identified, and some of which show cross-reactivity against asexual blood and liver stage of parasite development. These data clearly emphasize substantial physiological differences between asexual and sexual parasites, and the assay provides a tool for the discovery and development of transmission-blocking drugs. The productivity achieved with PerkinElmer high-content imaging systems can be exemplified by more than 1,000 publications between 2010 and 2019. The list of publications is available online and categorized by applications or research areas according to the imaging system used. Now I would like to introduce you to the latest high-content imaging systems from PerkinElmer. We have two models, Operetta CLS and Opera Phoenix Plus. Visually, you can see that they're quite similar in terms of appearance. And I would like to take this opportunity to share how we can image complex 3D samples like microbes tissue using the systems. Next, we would like to demonstrate what is the benefit of using confocal imaging together with the water immersion objective when we are handling complex samples such as a 3D microtissue. So shown here are some examples of images of the same liver microtissue stained with cell mass red and Herx, and was acquired with either an air objective or water immersion objective in wide field imaging on the top panel. So clearly you can see that there's a lot of signal scattering across the samples to result in a blurry image, and we could not visualize how this actual sample looks like. So typically we'll apply confocal imaging for thick samples to remove the out of focus light. However, there are some differences between the resolution when we are using either the air objective or the water immersion objective. Clearly you can see here the resolution is greatly improved when confocal imaging is combined with water immersion objective. So you can also generate movies with harmony. Shown here is an example of a plain movies or in 3D rotation. So these illustrate the software capabilities of Harmony. The workflow for image analysis is similar in terms of 2D or 3D data. First, you input your image data, and then you find your object of interest, followed by segmentation, and then you define your region of interest before quantifications on the different parameters of interest. Shown here is a summary of every steps in the cellular imaging workflow, whereby it starts from sample preparation and staining, followed by image acquisition, and then data analysis. And the imaging reagent was one of the missing pieces of puzzle within PerkinElmer portfolio. And we are proud to say that we have recently launched a new cellular imaging reagent portfolio known as PhenoVue to provide complete solution for the cellular imaging workflow. Now I'd like to introduce you to PhenoVue, our new range of cellular imaging reagents. We have a total of 27 reagents under the PhenoVue brand as of now. The cell painting kit is our flagship product, which will be offered in different kit sizes. The second category comprises of 14 organelles and cellular compartment dyes. And finally, we'll be introducing 12 fluorescent secondary antibodies. Shown here are some example images of the various organelles and cellular compartment, such as cytoskeleton, mitochondria, cellular membrane, nucleus and nucleoli, as well as lipid vesicles labeled in different color by the dyes. With that, I thank you for your attention, and feel free to contact us if you need any further information.